Hello, my name is Almeida, and today I will be talking about my news article, Prime Editing, the DNA tool that could correct 89% of genetic defects by BBC News. This article is related to our semester in Bio 282 regarding DNA translation and transcription we have been learning about, and the function of the synthesis genetic information to how mutations occur within an organism. Certain appearances of mutation can be harmless if the error resulted in the synthesis of the intended protein. In more serious mutations, though, the effects of the error could be detrimental to the health of the organism and can cause serious issues within the body. Certain conditions and diseases are associated with mutations of specific parts of chromosomes, and errors result in expression of those conditions. Examples of this include sickle cell anemia, Tay-Sachs disease, cystic fibrosis, among many others. Since these disorders are caused by errors in the individual's genetic information contained in their DNA, hypothetically, correcting mutations in the coding of the strands could result in the repression of genetically linked conditions. The target of this presentation is to analyze a news story regarding a scientific breakthrough in DNA mutation targeting and correction, and comparing the information to the primary research article. To better understand my news article and the primary scientific literature in which it covers, I will be describing some basic information about mutations that can occur within organisms' DNA. The expression of mutations can range from a silent mutation, which does not affect the individual, anywhere from a deletion or insertion, which can be crucial into the health of an individual and can result in the expression of a chronic illness or disease. As you can see, there are 4 base pairs in the DNA: adenine, cytosine, guanine, and thymine, which pair up to make amino acids, which result in the expression of a protein and eventually a characteristic in an individual. One change that can occur in an individual is a silent mutation substitution. This could occur here in the alanine. A substitution from the amino acid, which is guanine, cytosine, thymine, can change to ACU instead by changing this, which results in the exact same amino acid alanine. This would result in no change in the individual, and they would experience no symptoms of this. An example of a deletion that could occur in this DNA strand could be the complete deletion of the valine. This would be bad for the individual, as it would result in a different protein expression. The same could be said for insertion, as you could insert UGA sequence before the valine, which would result in the termination of this DNA sequence. This would also result in the expression of a different protein, as the DNA would be cut short. One other mutation that could occur could be a substitution that would also result in the expression of a different protein. This could be in the valine. If you change the GUC to a GGC, this would result in... Firstly, I will be talking about my news article, Prime Editing, DNA tool could correct 89% of genetic defects. This was posted on BBC News and written by James Gallagher. Firstly, my article spoke about previous DNA editing technology that is similar to prime editing. The most similar editing tool for DNA is CRISPR-Cas9, which can be used to edit DNA sequencing by identifying a target sequence, then cutting the targeted mutation in two. This causes some common issues not present in prime editing, such as issues with DNA sequence correction, as well as cuts ending up in the wrong place. This has created some controversy recently, as doctors in China created babies in lab that were protected from getting HIV. This raised ethical questions about the practice of CRISPR-Cas9 editing. The article was then followed up by a description of prime editing, the innovative technology. It is used to edit genetic code and to solve problems such as sickle cell anemia and Tay-Sachs disease. It has been proven to be more accurate than CRISPR-Cas9 technology due to its ability to both insert, delete, and substitute single letters of a DNA sequence correctly. The DNA sequencing is created in the lab using RNA, and it is meant to target the section you would want to make and create a change in the section. Of the about 75,000 different mutations which result in the expression of genetically linked diseases, prime editing is estimated to be able to fix around 89% of them. My BBC article seemed to correctly sum up the highlights of the primary literature article, which is titled Search and Replace Genome Editing Without Double-Strand Breaks or Donor DNA, but it left out some crucial information about the scientific processes of this technology. One important information is that it writes new genetic information using an impaired Cas9 endonuclease, which is similarly found in the CRISPR-Cas9 technology. This is also programmed with guide RNA, which specifies the target site and encodes the desired edit, as mentioned before. This technology was successfully performed on 175 edits in human cells, including insertions, deletions, and 12 types of point mutation substitutions. The technology was effectively used to correct mutations characterized by sickle cell disease and Tay-Sachs disease as well. Not only was this proved successful in human cell lines, but also is proved successful in post-mitotic primary mouse cells, which suggests implications in not only humans but animals as well. And then here's my works cited.